I build applications and web tools and R packages, cloud deployments, just a few things here and there. So, okay, great. So we've gone through this pipeline where we've been able to log transform our data and do normalization and do statistics and make educated choices about all of those things. But as soon as you get in the weeds, when you're doing science, it's very hard to frame the problem where you go back to the big picture. What are you trying to understand about the system? What is your question you're trying to answer? So here, what kind of questions are we trying to answer? Are we trying to answer whether there are trends in peptide protein abundances or fold changes, or in cases of other data sets like metabolites, am I interested in seeing if amino acids, for example, have changed expression throughout time? Am I interested in understanding the differences of certain peptides or proteins between soil samples that have been under drought and re-wet conditions? What am I interested in seeing or exploring? So here's where you might take the data, the statistics, the normalized abundances, and try to come up with some visualizations that are easy to explore and easy to use so that you can answer these questions. Even when you take those, 250,000 peptides and then roll it up to the 100,000 proteins, that's a lot of data to look through and to identify patterns. If you try to throw that into perhaps a ggplot with the facet wrap for each individual plot, you're not going to be able to see all 100,000 ever. In fact, people that manage to get 40 plots in a frame do a really good job, because that's difficult on its own. So here we're going to cover some tools on how you can actually visualize this complex data. Maybe perhaps you're interested in the predictive power of your peptides and proteins or metabolites. We mentioned what's different between groups, but maybe there are some biologically significant things that you aren't capturing with your statistics. So you want to use perhaps a different model, or if you've got a bunch of time periods, doing every comparison of every time period is probably not your best approach. You might want to instead look at a regression model. So maybe you don't want to do the exact statistics that we presented at the end of this pipeline, but you want to take that normalized data through PMART up to that step and then run a model with it. Or maybe you don't even care about the differences in the groups. You just want to see some dimension reduction and see if you can identify patterns in between each of your samples. Maybe instead, too, you're more interested in the interrelationships of peptides or proteins or significant subsets of each of these between groups and within groups. And then enrichment analysis is also something people tend to do with this kind of data. That's when you take either your significant peptides or proteins for some algorithms. In other algorithms, you can actually take the fold changes or the normalized abundances and use that data to then see, okay, which sets or which groups of functions across my protein data are most significantly different between each of my two groups. Or perhaps pathway analysis. We didn't really cover this here, but there's lots of other types of proteomics. I know, we talked for an hour, we covered a lot. There's even more. You might hear things like phosphoproteomic technologies or glycoproteomic technologies, which will have their own specific sets and questions to answer. And so those require maybe different tools with different assumptions. So here we're just going to kind of give you a high level view of what people tend to do with this data and which kind of data they use for each of these. So let's say I'm just interested in understanding more about my biomolecule abundances and my fold chain. Back to when I was mentioning, we've got large matrix data is how I think about it, right? We've got lots of different plots to look at or panels. And that's where you can use things like Trelloscope. So within the PMART package, when you finish your normalization step, you can actually wrap it in what's called a Trelli data object, which is a Trelloscope-based object where you can take that information, pass it to a summary function, and get a summary of which kinds of plots you can make. So for example, if you've got omic data, you can make a specific plot for each omic. So if that's a peptide or a protein, you'll get one of these plots for every single peptide and for every single protein. And then you can look at more information like your specific groups of comparison. You could also look at it across classes. So if you've want to see the expression across proteins, You can then get a plot for every single protein and see, okay, across my groups, can I find something of interest? The nice thing about interactive trelliscope displays is they allow you to have cognostics where you can sort by p-values. So you can at least start there for things of interest. But as we always know, biological significance, statistical significance are not the same thing. And the 0.05 threshold cutoff is arbitrary and everybody's ran with it. So there might be things that are within that borderline, even to 0.1 or 0.15 significance, that are important and useful. Thus, it's always good to just look at your data. You might find something interesting. We also have the abundance data itself as histograms, so you can start to see distributions for each of your omics of interest, your peptides, or for each of your peptides, we have these automatic heat maps for these abundances, where you can look at each of the biomolecules on the x-axis and each of your samples on the y-axis. And if you have grouping information, we'll automatically sort it for you, so you can look across each of your groups. Any of the statistics involved with these three plots, what do you think it's going to be? Do you think it's going to be the ANOVA statistics or the G-test statistics involved with abundance? You meant to think about it. Not a hard question, I promise. I have someone who wants to answer. So excited. All right.
Nova?
Yes, exactly. Because the G-test is going to take these missingness values as what it's testing for significance. So we do have a missingness bar plot here. You can look at missingness across each of the omics, whether that omics is involved in the abundance, or you could just pass it our statistics result object, which is what this is telling you here. And you can look across each of the biomolecules, biomolecule classes, and samples. If you're interested in the statistics, it's only across each of the biomolecules, right? So that's going to be where it's most useful to visualize the data for those filters. The entire point, by the way, as I'm going through each of these differences, to give you an idea of things you can look at and do when you get your results and you're like, I don't know what to do with this. This is to help you look for things that you know, look for that protein of interest, which you can do in the telescope display. You can type in the exact name of that one protein that your PI cares about. You can go find it and pull that plot real quick, and then maybe also show other associated proteins as you search around different biological databases. It's here to help you. We built this to help you reach these biological interpretations after statistics. So here we've got a bar plot, which is show each of the fold changes. So you can see between each of your groups where those change. Anything significant will be highlighted with a bar around it. And then we also have box plots, which you're going to need to use some sort of molecule class information like the proteins, because to get an actual distribution, you need more than one sample, right? Each of these is just showing you one because we're on that biomolecule level. And as soon as you have more information for groups of biomolecules, like in those eMeta or those biomolecule information files, then you can actually start to visualize data on that level. We also have everyone's favorite, the fold change volcanoes for each of your comparisons of interest. So if you have multiple comparisons, like here I'm having a group 1, group 2, group 1, group 3, group 2, group 3, it will put all of those in one telescope display for you, and you can place individual panels next to each other, visualize multiple comparisons at once for each of your, once again, you're going to need classes because you're going to need more than one biomolecule. And then we have a heat map once again, but in this case, we're looking at fold changes, not abundances, per comparison, not per biomolecule, and across your biomolecule. So it's a very different plot. So here I've got an example of a screenshot of MODE here, the multi-omics data exploration tool, because Lord forbid everything doesn't have an acronym. That's what it's built for. And when you're looking at here at this little disco ball, we're trying to get at this point of these matrix plots I'm telling you about, or a trellis. That's why they're called trelliscopes. I don't think anyone ever directly says this, but when you're looking at a trellis where the plants grow up, each of the rings, each of those little squares looks like a plot in this kind of design. So here with Trelliscope, the nice thing is if you use the PMAR application, which we will do this afternoon, you can export your normalization results, click one button, everything before this will autofill for you. So you never need to specify anything again. You need to take that information and then go to which kind of plots you want. So here we've just got that first plot, that abundance box plot I was telling you about. These are the abundances. Each of these is a group of samples. So that's what we're looking at here. And we have options to specify changes to your ggplot here. You select your cognostics. Those are the things I keep coming back to where you can sort data on. You could sort by maybe your number of samples, mean abundances between each of these. They're box plots, so median's actually more useful. That p-value, so here that'll be that ANOVA p-value. Thank you, Ava, gold star for the day. And once you're all done, you just type in a name for your telescope, you click create, and then you have a standalone HTML telescope display you can explore. You can also take ones you've made and load them back in the application to then explore patterns across abundances, missingness, and fold change. So here's a way that you can try to reach conclusions with your data on your own and be empowered. And the nice thing about these shareable and sortable telescope displays is you can also send that to your advisor, teach them how to use it, and show them how you found interesting things in the data. They're very nice. Okay, great. So I kind of just took us through one way that we can answer a bunch of questions with data exploration. Maybe here you're interested instead of understanding the predictors or your target protein for a specific group. So one way people try to understand their predictors in terms of their biomolecules is to use supervised machine learning or prediction. So here we've got a very simple random forest, I suppose. It gets to some number of trees where we're taking across each of our biomolecules, which is going to be our input predictors, where we're using those normalized abundances. What is the response in this case? And our responses are going to be those phenotypes of interest. So whenever you fit these kinds of models to answer your question, there's a scale that you should be thinking about in terms of interpretability and model usability, right? So like some deep convolutional neural nets might have accuracy, but you might not be able to pull the nuances, those beta coefficients of which of those biomolecules are most informative for your decision, right? At least with random forests, we get some interpretability because we get importances, but as soon as you do regression with a random forest, you lose the actual coefficients from those beta values. So there's kind of that scale that you should talk to a statistician about when you're trying to fit models to know what to use, because there's a lot of information to track, which seems to be a theme of all of my talks, is that there's a lot of information to track always. So for example, if your output is categorical, you've only got two, thus the binary, you could use logistic regression. You do get some closed form equation and you do get variable importance. But as soon as you have three groups, which is very reasonable to have more than two groups you're looking at, you can no longer do logistic regression at all, and you lose perhaps a very highly interpretable method. If you got random forest, you could do a multi-class, but once again, I was mentioning you couldn't have those beta coefficients. You don't end up with a closed form equation. You can't just predict based on a specific equation. You can do predictions, but you might not understand why things go certain ways. And you do end up with variable importances there. For linear regression, you're going to require some sort of continuous output. So here, that's a case where I mentioned if you've got several days across an experiment, you probably don't want to do comparisons, because as soon as you get to 7 comparisons, that's what, 7 times 6 total. So that's 42 plots. And as soon as you give your PI more than 2 plots to look at, they're probably not going to be happy with you. So you're going to have to be very careful with what you show. So maybe instead with this data, you want to take those input normalized abundances and do some sort of linear regression with it, and have your output be that continuous value. You end up with that closed form equation. I feel like linear regression is very straightforward, and all of us have been doing some form of it since like pre-algebra. Remember when you had to draw lines with colors back in the day? So it's not too hard to understand fitting these kinds of lines. And once again, you only get variable importances, those beta coefficients, if you've got some sort of standardization for them. Then we start to move into other algorithms, things like K nearest neighbors, which is a categorical one, but this is one where you need to know how many groups you're supposed to be looking for. You don't get a closed form equation and you don't get variable importance. And we get classifiers like naive-based classification, or just you could do a single decision tree. That's another one where you've got continuous inputs or categorical in terms of a singular decision tree. You do get a closed form equation, but you don't get a variable importance. And it goes on and on. I made this a very short table for a reason. So once again, this is a lot of information to track, which is why we have a push here at PNNL to help design applications so that you can start to answer specific questions about your data, specifically here with omics data, have an informed decision about what kind of model do you want to fit. Questions like, we'll read your data input and we'll ask, is it categorical? Is it 2 categories? Is it multiple? Is it continuous? Because already that first vital piece of information is going to remove specific things you can do. Then after we remove models that are not a good fit, we can rank them based on things you care about. Do you care about closed form equations? Do you care about variable importance? Your PI might tell you, I don't care about those things, until you get a really good classification algorithm. And then they ask you, well, how does it work? And you say, I don't really know how it's working. And I don't know about the interrelation. And that gets us back to the interpretability piece, which is really important. I think I'm going to just actually pause for a second, see if there's any questions on maybe fitting equations, the kind of decisions we make for those. If anyone has any questions about their own data that they're trying to fit, maybe we can talk about it. So I'll give us like a minute. Yeah, we got a question. Do we have someone passing around?
That's right.
Perfect.
Yeah, what's a support vector machine?
Yeah, so this is another classification algorithm. It is technically binary category wise, but what the R package won't tell you is that it will split it several times, but it takes basically a scatter plot, looks for your group, and then applies a spline or a division between each of your groups. And so you could fit this with linear regression for a straight line. It also does radial lines, so circles in between each of your groups, etc. So it is a way to define a boundary for each of your groups of interest in a two-dimensional.
When you say it'll split it multiple times, do you mean that you can throw in more than 2 and then it'll hierarchically split them, or?
Right. On the mathematics side, if you care about that, it's applying 2 at a time. And so it will do actually a loop for each of those, and you'll get a bunch of model outputs because it can only split 2 at a time. But in the R package, you can ask for 3 or 4. And if you don't know what's going on and you do know it only does 2 at a time, it will freak you out a little bit. So that's what's going on the back. Yeah, you're welcome.
We have a question online. How do we decide on which algorithm to use?
So once again, there are some things we're working on where we're actually just defining what each of these categories are, because I find tables like this very helpful. And we have a table that is way longer than this that we're constantly adding to for different things that people are interested in. So once again, the famous Kelly answer, it depends. It depends exactly on which kind of input data you have, what you're trying to do with it, how interpretable you would like it to be, etc. And just know that like, this is why engaging statisticians early and often is a good plan. Oftentimes, you know, you might not be able to fit a linear regression model, but maybe some sort of decision tree can actually do the job for you. Or maybe you just need an anomaly detection algorithm to use, and that's good enough. There's so many options. Here, we're just kind of sticking with traditional statistical learning things that you can run with small data sets, which is what we have for proteomics and metabolomics at this point. We don't have billions of lines of data. We don't have all of Twitter to learn our data sets on. We have these smaller data sets, which is why I've hand selected just a few of these models. I also think in the chat, I talked about this a little bit. A good friend of yours is Tidy Models. Just look up tutorials on Tidy Models and how to fit these models real well. Didn't cover it here, but there's lots of stuff you do in terms of cross-fold validations or testing and training data sets where you hold out your testing data set and try to improve that with both that cross-fold validation approach. There's lots of ways when you fit a model to make it better. So if you fit a random forest and you get an okay value as your accuracy, don't panic. Go look at these packages like Tidy Models and see if you can't improve the fit of your model, because it might not actually be that bad. Okay. So I think another thing that people are interested in when they get this kind of data, besides fitting perhaps different models for specific questions or visualizing those abundances, missingness, and full change, to maybe understand in a relationship, either across different omics types, which is integration, which on day 5 we'll cover some specific methods for, or within your own data set. So people tend to use things like correlation matrices, and maybe you're interested in this information within a protein. Maybe you're interested in looking at how peptides correlate to other peptides within your data set. Or if you have metabolomics, maybe your specific metabolite class. And so the kind of correlation you use matters. We had some really good questions earlier about parametric and non-parametric tests. Also, things you need to consider is whether the data is normal or not. If it's non-parametric, maybe you want to use one of the many non-parametric correlations, which includes like experiment options. Another thing people tend to build are interaction networks. And this is interesting because when I first started, I understood the world of functional enrichment and set enrichment, where you have for each of your proteins that shared family of functions. Tyler is going to share a little bit more about, and you try to understand maybe differentially expressed functions. Because let's be real, a list of like 600 transcripts is only so helpful. You're trying to get to the meat and bones of maybe what's different between the systems. That's what that kind of enrichment does for you. Or maybe pathway analysis, which is a similar kind of approach where you look at the pathway itself and see which parts of the pathway are up expressed or down expressed between each of your comparisons. And then maybe above that, I think of, you got your sets, so that shared functions, you got your pathways, which are intense, biologically defined, understood networks of interactions between proteins, metabolites, certain RNA transcripts. You got it. It's very intense pathway analysis. Maybe there's a higher level, which is where statisticians like to think, which is just interaction. Does that protein interact with that protein in a pathway, in a set? In any way, do we have interactions between each of those proteins? And in your study, have you found something new? Have you found something outside of literature between two proteins interacting? So that's something interesting that people do. There's kind of two trains of thought for interaction networks. There's people that mine them from literature with natural language processing tools. And then there's people that pull them from places like KEGG. There kind of is sort of a schism, I suppose, between the two. The people that insist on manually annotating everything and being 20 years behind, and the people that insist on pulling stuff from literature, also knowing that in literature, lots of stuff gets published that shouldn't be published or gets published quickly. I know that in some studies we do, when we pull this kind of information, pull networks from papers, we tend to skip most of 2020 when it comes to COVID analyses. Does anyone know why we might want to just skip that year entirely? We have a brave one. You're doing great. You get another gold star today. Don't worry about it.
So I would guess it will be inflated publications.
Yeah, there were lots of papers published very quickly on COVID interaction networks, especially within those first 6 months, where lots of things were found, like studies with batch effect that weren't properly corrected, et cetera. And so a lot of times when we do analyses, we just blanket get rid of those years. So yeah, that's just something else to consider if you're doing the mining literature approach. Know what's going in there, know what's your literature you're pulling from and what kind of requirements you're using. So like I said, everything got its own pros and cons in biology. So that's one way people do it. And another way, we've mentioned this before, is perhaps you don't really know what your response is going to be. So you're going to do some sort of unsupervised machine learning with various clustering methods. There's lots of options for those too, and lots of reasons why you should and shouldn't use certain ones. I can give you a hint with the kinds of data that we use here, those normalized abundances with the peptides. They have lots of missingness. So whatever dimension reduction you use, make sure it counts for missingness. If you have one that doesn't, and you have to filter any peptides or something's missing, you might end up with 3 things you're trying to cluster at the end. So just be careful. Projection pursuit PPA is a good one to use. That one accounts for missingness, and it's built into the PMARD-R package. So all you have to do is take one of our objects, do dimension reduction, and you're golden. Yeah. So we'll wrap that for you. So that was kind of like a high level overview of lots of the things people tend to do. There's intense levels of detail that I cannot cover and will not overwhelm you with right now. But once again, I track that information, so you don't have to. So if you have specific questions about your data sets, about the kind of analysis you want to do, how to get into natural language processing, if you decide protein-protein interactions and novel discovery there is what you want to do, how to use the R telescope visualization, we're here for you. This is a growing area. We are constantly learning about it and adapting with our data sets to what is new and novel in this kind of biological interpretation world, as I'm calling it. So with that said, I'm going to introduce Tyler up here, who's going to kind of cover high-level enrichment analysis, not so much for transcripts, but specifically for the kinds of proteomics data sets we've been talking about today. So pass it over to Tyler.
Thanks, David. So we're going to cover what's broadly called enrichment analysis. It's also called gene set testing because the language was developed with microarray and transcriptomics data in mind. So we're going to look at how does it work? What's the broad definition of this type of analysis? We're going to look at the distinction between enrichment analysis and what's called overrepresentation analysis, because these are often used incorrectly interchangeably. We're also going to talk about some of the advantages of enrichment analysis, as well as some disadvantages. And I'm going to show it in action for a paper that's actually about to be published. So despite how it looks, this is the abridged version of this process. So the first step is we obtain some sort of biomolecular statistic. So we talked about the ANOVA statistic or the G-test. So the question is, let's say you have, I'm going to use genes interchangeably with biomolecules and proteins. So if I say genes, that's what I mean. So if we have a vector of, let's say, 10,000 genes, so we have our statistics for those genes. The question is, how do you determine biological meaning from that? Do you simply take the top 100 genes that are changing and figure out, okay, what's going on here, do a literature search? Or do you assume some sort of arbitrary cutoff, like David said, a 0.05 cutoff, and just say everything that passes that is interesting, and we'll look at those. So what we do instead is, because if we limit ourselves to that top 100 or everything that passes this cutoff, we're not making full use of the data. We're going to miss a lot. And not only that, but it's very time consuming. So you can imagine, let's say 50% of the genes are differentially abundant. So the problem is, are you really going to sit there and go through all 50% of those genes, do a literature search, try to figure out what's going on, what their interactions are? You're going to miss a lot of information. So with this, we have our vector of statistics. We're going to sort them from highest positive to most negative. And then from there, we're going to calculate an enrichment score and figure out, okay, is this significant or not? So the process for that is where I'll explain in the next slide, but we're calculating an enrichment score that describes a group of genes called a gene set. So this could be a group of genes that's involved in the same pathway, could be located in the same area of the cell. They could be related in some other way. It could even be a set of genes from a prior publication. So you could say, they found this was interesting. I want to take a look and see if it's also interesting in mine. So we're going from these gene level statistics to these gene set level statistics. So we're summarizing. So it's a lot more digestible and easier to go. So the next step is, okay, we have this enrichment score. That's basically saying we have like a T statistic or an F statistic. Now, that doesn't really tell you much by itself. It'll tell you, is this set primarily upregulated? Is it primarily downregulated? But you don't really have an idea of whether that's actually important or not. You have no sense of the scale or the significance. So the next step is to permute the labels for your original vector of statistics. So now you generate what are called permutation enrichment scores. So you might do this 1,000, 10,000, 100,000 times, and you end up with a distribution of enrichment scores under the assumption that there's no relationship between these scores. That is, that the genes in the set are randomly distributed throughout your ranked list because of this permutation. So from there, you can simply define the enrichment p-value, and it's actually a little more complicated than this. You can define it as the proportion of these permutation enrichment scores that are as or more extreme than your original score for a given gene set. So now we have enrichment scores, we have our p-values for each gene set. And what we need to do next is just adjust them to account for multiple hypothesis tests. So the way to do this is you can just use the procedure Benjamini and Hochberg is typically what's used. Anything that would control for the family-wise error rate is probably going to be too stringent, so I would avoid that. So anything that controls false discovery rate, I would encourage. So here's a way to sort of visualize what's going on here. Button is the top one. Okay. So at the top here, we have an example where the genes in a particular set S behave randomly. That is, the genes in blue are distributed pretty uniformly throughout this ranked list. So here we have, I should explain that first. This plot is showing the y-axis would be whatever statistic you have. And then the x-axis would be the rank of that gene. So we rank them from highest to lowest. And this is what the input into this method looks like. So here's an example where they're randomly distributed throughout, so not much is really going on. We can't say that a gene set is primarily up regulated or primarily down regulated here at the bottom. So this is also what your permutations would look like. So if you're randomly permuting the labels, the genes would be distributed throughout like this. So this is what permutations would actually look like. And then these are examples of sets that are primarily upregulated and primarily downregulated. And you can see within a set, you're allowed to have genes like these two at the bottom on the left side that are actually upregulated, where most of them are downregulated. So we're just trying to summarize the behavior of the set. We're not necessarily trying to say all of the genes in the set are up or all of them are down. So we just want to summarize and then get an idea of how significant that actually is. So the procedure for generating this enrichment score, you're probably wondering about what are these black lines. So what you do is you start at the most positive statistic and you say, is this gene in my set S, particular set S? If so, you're going to increase a running sum by an amount that's proportional to that statistic. So if it's near the ends where the statistics are more extreme, then this running sum gets increased more, and it's using the absolute value. So you can see here, the panel at the bottom, it's negative because it's not really encountering genes in the set. And then all of a sudden it starts to encounter a bunch of them. So it starts to increase, and you can see it actually reaches 0 at the end. It may or may not do that. So the enrichment score is just the maximum deviation from 0 of this line. So you could have a line that goes up, and then it goes back down, and then it's down again at the end. So that might mean half the genes in the set are up, half of them are down. And you can't really say what's going on with that set. Or you might have cases like this where it's very nice because this is simulated data, so it looks very nice, but that's not always the case. Where it's very clear that the gene set is primarily up or primarily down. So I'll pause here if there are any questions. I know I threw a lot of information. I'll assume it was crystal clear then. Clear as mud, right? So I want to cover the distinction between the overrepresentation analysis and the hypergeometric, enrichment analysis. So overrepresentation analysis, it uses the idea of typically a hypergeometric test. So I like to use the example of M&Ms. So let's say you have a bag of M&Ms, and if you take a handful of some size, what's the probability that in that handful, let's say you have a handful of size 20, what's the probability that half of them are blue? Well, you probably think it's low unless your bag of M&Ms is 50% blue. So you might say, hey, he's fishing for blue M&Ms, and you're probably right. So this overrepresentation analysis, what it's doing is you impose some sort of arbitrary cutoff, let's say FDR of 0.05, and you say everything that passes this threshold, I'm going to call it interesting or significant. So this is your interesting subset. And then you have your overall population of genes from your experiment. You have your subset and your population. Then you look in that subset, and for a particular gene set, you say, is this gene set more present in this small subset than what you would expect just by random chance? So that's usually meaning that if it's localizing in the subset, the genes in the set tend to be either upregulated or downregulated. They're significant in some way. And again, we're using this arbitrary cutoff, which is one of the drawbacks because you're not making full use of the data. And also notice that we're only saying whether something's significant or not. We're not really caring about direction. So we're losing that information. So what if a gene set is primarily up or primarily down, or it's a mix of both? You have no idea. You could separate the data into positive and negative, but again, you're kind of losing information. So what we would say when we have a result, we would say this particular gene set is overrepresented in the subset of interest. So on the other hand, enrichment analysis is the p-values are permutation based, so we're not doing any parametric tests. So it's very slow, unfortunately. One of the benefits is that it uses all the biomolecules in the experiment. So we're making full use of all that data that we spent all of our time collecting and all of this efforts that we really want to make use of. We don't want to just say, yeah, 0.05, it's good to go. And then we're also making use of the direction and the magnitude of change. So before, if something was significant, it was significant. It's just binary, yes or no significance. But here we have more granularity, and we have an ability to actually detect sets where the genes are changing, but they may be very small changes that aren't significant on their own, but overall the set is displaying some sort of concordant behavior that might be interesting. So making full use of the data, able to actually see more of what's going on. And then the language, you would say a particular gene set is positively enriched or it's negatively enriched. And there's no subset. So you just say it's positively or negatively enriched in your data that you have. So even though ORA has a lot of drawbacks, in some cases, it is better than enrichment analysis, or it is your only choice. So if you, let's say, take your data and you cluster it, then you might say, are there any sets that are more present in a particular cluster? Or you can run this for all the clusters. So here, you can't really use enrichment analysis because you just have in the cluster or not. That's your only information. So in that case, ORA would be actually the only option. So as we kind of talked about, enrichment analysis, one of the benefits is that it doesn't rely on this arbitrary cutoff, so you can make full use of your data. The other thing is you don't really need biological knowledge. This is also true of ORA, but we're making use of existing biological knowledge. So like me, I don't know anything about biology, to be honest. So having a method where I can just say, okay, the experts in this field have curated these sets. They've spent a lot of time figuring out what pathways, what genes are involved in certain pathways. You can make use of that data. And it's also very reproducible that way. So if you're a biologist and instead you're trying to do this manually, going through that list and figuring out what's interesting, you're going to be unfortunately biasing your results, whether you think you are or not. So you're going to be searching for things that you think are going to be happening, or your knowledge is going to cause you to miss some things just because you don't know. Everybody's limited in what they know in some way. So the most important thing I want to talk about here is that I've used the language of genes and gene sets, but you can generalize this procedure to any biomolecular set. So you could have phosphosites and kinases. So you could have substrates grouped by the kinase that phosphorylates them, and you could figure out sort of indirectly, are these kinases active or not? And whether they're up in one condition or down in that condition. So your use of this is really just limited by the data, the sets that you have. So don't think about it as gene set enrichment analysis, or think about it more as biomolecular set enrichment analysis. And also, it may have uses outside of biology. So that might be something to keep in mind. So I've talked about these gene sets or pathways. So here's an example. What people typically use is the gene ontology database. So the gene ontology database is broken up into 3 sections. So you have biological processes, cellular components, and molecular functions. Each of these is organized in this sort of tree where each term on a lower level, where you see the arrows connecting, is a subset of the term above them. So as you go down, you get more and more granularity. You get more specific terms. So you see at the top, it always originates with metabolic process, biological, I forgot, biological process, molecular function, or cellular component. Those are the root terms. So if you want to learn more, you can look up the documentation for the gene ontology, but this is one example of gene sets that are available. And they're also available in the molecular signatures database, MSigDB at the top there. So here's an example of using this, what's called FGSEA, so fast gene set enrichment analysis. So here, this is, I believe, biological processes. It's one of the gene ontologies. So what you do is you end up with these, what are called normalized enrichment scores. So it's like the enrichment score, but you're just accounting for the size of the sets. So if a set is very large, then it's usually going to be more enriched than smaller sets, just because there's more chances for that running sum to increase. So we're just accounting for that. So anything within 1 and -1 is not interesting for reasons that I'm not going to get into, but for this particular method, we're only concerned about things beyond those bounds. And we can see that each of these terms is colored by their normalized enrichment score. So you see a lot of terms in blue indicating that they were, that they had primarily negative correlation, because here this is correlation based. So primarily negatively correlated with some metric of the samples, but we have one term that was positively correlated. So it might be interesting to look at these terms and look at the members and see what's actually going on. So rather than looking at all of the genes in the data set, you can kind of narrow down to just a few terms that look particularly interesting. And these have a nice descriptions as well. So you're not trying to, you're not relying on biological knowledge that you may or may not have. So here's a more complicated example where we have, at the bottom here, we have each comparison. It's like we're doing a bunch of t-tests, basically. So we have 1 week trained, 2 week trained, 4 week, and 8 week trained rats that ran on a treadmill. And we're comparing them to a group of sedentary rats. So what we're trying to see is whether treadmill training affects any gene sets. So we have all these comparisons, and we have females and male rats, and we can perform FGSEA separately for each comparison, and we end up with results that we can then plot in this heat map format. So what this is showing is, let's say for the male rats, you have all of these terms at the top on the left of translation initiation factor, ribosome binding, translation. So you might say there's something going on with translation. So I can look into that more. And this also might be a indicator of a lot of overlap in the terms, just because the gene ontology databases, since we saw it's hierarchical. So you can have a lot of redundancy, fortunately. So, or here in females, we can see that this structural constituent of muscle term at the bottom actually decreased with 1 week of training, and it continued to decrease at all weeks, but it was less of a decrease at 8 weeks. So we can see actually the temporal pattern of this set. And then on the right, we have an example of where this was applied to metabolomics and lipidomics data. So we can see we can group metabolites or lipids by their chemical subclasses. This is from the RefMet database. So we can see like acylcarnitines were significantly up with 1 week of training relative to the sedentary group in females, and also in males, though the degree of change wasn't as pronounced. And we can see everything that's actually significant is being shaded with this gray background. So you can easily pick out, okay, what's actually changing. And you can see over time, like here, OPE term was significant in the 4 week versus sedentary comparison, but then it drops off at 8 weeks. So it's appears very close based on size. So the other things you need to be aware of when you're dealing with this type of analysis is set redundancy, which I kind of covered when we talked about the gene ontology database. So you have a lot of terms that maybe share genes, or genes are typically involved in many different processes. So you have redundancy and you have relevance to your experiment. So if I have data from, let's say, brain tissue, I don't probably want to be testing muscle-related terms. That's not really going to tell me anything. So you want to really limit your sets down to what you think is going to be interesting. Sure, you can kind of throw everything at it, but be aware that you're going to have to adjust across everything. So you may lose your ability to detect those actually interesting changes when you throw in a bunch of irrelevant kind of garbage. So the other thing is the size of the sets. So you have a trade-off between reliability and specificity. So as you go, as sets get larger, they get more reliable, because a change in one gene in the set doesn't really change the overall enrichment score of that set. But the problem is it gets really hard to interpret. If you have a gene set that's consisting of 50% of the genes in your data, like, good luck. You're not going to really get anything out of that. On the other side, on the other side, as sets get smaller, they get less reliable. Again, as one gene changes, it can affect the set more strongly. The other thing is mapping between identifiers, which I'll you probably already know is a huge issue. And then your choice of ranking metrics. So I talked about t-statistic. You could use the g-test, you could use the ANOVA F statistic, but be aware of what you're using and how that may affect the results. So here, I don't expect you to take notes or write this down or anything. So once you get these slides, you can look through all these resources and just summary of everything, but mainly it's growing area. Things are always changing. There's a million of these methods out there. I only covered one, and it's not necessarily the best. So you need to make sure that you have a good understanding of these methods and that you're not contributing, like David said, to the garbage out there in publications.